Hello, this overview covers eukaryotic cells. There are two main types of cells, prokaryotic cells and eukaryotic cells. Prokaryotic cells were reviewed in another video. Eukaryotic cells are cells that have a membrane-bound nucleus which contains the genetic material and controls the activities of the cell. They also possess many membrane-bound structures called organelles. Eukaryotic cells are found in the domain Eukarya. Eukarya is divided into the kingdoms animal, plant, fungi, and protist. The nucleus contains the cell's DNA and controls metabolism. When the cell is busy carrying out its normal functions, the DNA is stretched out in sections, unzipped, and being read. When it is in this form, it is referred to as chromatin. Also in the nucleus, you will find round gelatinous structures called nucleoli. There can be one or more in a nucleus, and it's in this area where the subunits of ribosomes are made. The nucleus is surrounded by a double membrane, and the two membranes are connected to each other. It is referred to as the nuclear envelope. The envelope contains pores that are made by proteins. These pores allow for the passage of messenger RNA and other proteins. Ribosomes are made out of ribosomal RNA and proteins, and they come in two subunits. Think of them as hamburger buns. There's a smaller bottom piece and a larger top piece. The two subunits will fit together on a messenger RNA to read the messenger RNA and make proteins. Ribosomes can be found floating freely in the cytoplasm or attached to internal membranes. They are usually represented in cell models as dots. Endoplasmic reticulum, abbreviated as ER, comes in two types, rough and smooth. The rough ER is studded with ribosomes and looks rough. The smooth ER is more tubular and has no ribosomes, so it looks smooth. Rough ER is responsible for producing protein products to be held in vesicles within the cell or released out of the cell. Smooth ER produces lipids and carbohydrates, stores substances such as vitamins, and does the job of detoxification. The Golgi, also known as the Golgi apparatus or Golgi body, consists of a set of flattened membranes that are interconnected. They collect, package, and distribute molecules that are delivered from the ER. Vesicles will pinch off from the ER and travel to the receiving side of the Golgi, known as the cis-face. The Golgi will modify the products, tag them, and redistribute them. The products exit out of the transface of the Golgi by pinching off in secretory vesicles. The ER and Golgi are the major components of the endomembrane system. One of the products of the endomembrane system is a structure called the lysosome. Lysosomes contain hydrolytic enzymes. Remember that hydrolysis breaks monomers apart. They are basically the stomach of the cell. They digest old, worn-out structures and materials taken in by the cell. Another product of the endomembrane system is the peroxisome. Their major function is the breakdown of fatty acid chains. Hydrogen peroxide is a waste product produced when you break down the fatty acid chains from triglycerides. Hydrogen peroxide is very harmful to a cell, so it's immediately broken down into water and oxygen. Larger vesicles that isolate substances in the cell are known as vacuoles. Vacuoles are most prominent in plant cells. They function in water balance and storage of water, salts, toxins, and pigments. Single-celled aquatic organisms regulate water balance by squirting water out through contractile vacuoles. Vacuoles can be found in adipose cells where they store fat. The mitochondria is the site of cellular respiration. It produces the ATP energy molecule needed by the cell. There's an outer and an inner membrane. The material inside the inner membrane is known as the matrix, and invaginations or folds of the inner membrane, known as cristae, increase the surface area for enzymes and proteins that produce ATP. Also, there is an inner membrane space between the two membranes, which is important in the function of the mitochondria. Mitochondria also contain their own circular DNA and ribosomes, which means they can make some proteins. The cytoskeleton helps maintain cell shape, assists in movement of the cell, and transports material within the cell. The cytoskeleton is very dynamic. It can be assembled and disassembled rapidly. It is made up of 3 protein fibers. The first are called actin filaments. They're the smallest, and they're made up of a helix of 2 actin chains. They're responsible for contractions and crawling. Interactions with motor molecules, such as myosin, allow actin filaments to slide back and forth, causing muscles to contract and relax. It is also the same fiber that allows the amoeba to ooze along the microscope slide that you observe in the cell lab. The second type of fiber are called intermediate filaments because they are intermediate in size between the other 2 fibers. Their function is to support the nuclear envelope, to create cell-to-cell junctions, like holding your skin cells together, and to provide strength to things like human hair. The third type of fiber are called microtubules. They are made with proteins called tubulin, and they organize and move materials within the cell. Their assembly is controlled by structures known as microtubule organizing centers, or MTOC. The most important MTOC is called the centrosome. Vesicles are transported along microtubules using motor proteins. There is a link in Blackboard to a video done by Harvard University which shows microtubules moving vesicles inside a cell. You can see these motor molecules walking a vesicle along the microtubule. Centrioles are found inside the centrosome of animal cells. They function in cell reproduction. They are short cylinders of microtubules in a pattern called a 9+0 pattern of microtubule triplets. There is a set of 3 short microtubules known as a triplet, and they're sort of glued together. They are linked with another triplet by motor molecules. There are 9 of these triplets connected in a circle, and the 0 refers to the fact that the space inside the centriole is empty. Flagella and cilia are external structures involved in movement. They are hair-like projections covered by membrane, and they have a 9+2 pattern of microtubule doublets. So instead of 3 microtubules linked together, there are 2. There are 9 sets of those in a circle, and there are 2 microtubules in the center. Cilia are many short projections that usually occur in rows, and they move the cell through a liquid environment, or they move things across the surface of the cell. We have cilia on the cells that line our upper respiratory tract, and they move mucus up out of our lungs to clean the air that we breathe. Flagella are long, whip-like, and much fewer than cilia. There's 1 to 5 of them attached on one end of a cell. Flagella move like a propeller, while cilia move back and forth. Animal cells may include an extracellular matrix secreted by the cell that functions to provide support to the surrounding cells. The extracellular matrix is very important to the function of bone and cartilage. Please watch the plant cell overview video for a more complete picture of cell structures and functions. Also, go to Blackboard and watch the Inner Life of the Cell video by Harvard University. It shows an animation of the interior of a white blood cell, and you should be able to recognize most, if not all, of the cell structures. Thank you for watching this overview.